Okay, so the examples that I've shown so far, I just had one time series of data and one state that I was trying to estimate. But in the real world, oftentimes we have multiple streams of data. And those multiple streams of data are helping us giving more information about this underlying state that we're trying to estimate. So here's a couple concrete examples. So what if my goal was to measure temperature in a region, and I had multiple stations where I'm measuring temperature. These happen to be multiple personal weather stations in a fairly small area. And let's say in this valley, we might imagine that these are all close enough together that we think that they're measuring the same temperature, but they're all a little bit different because there's, the micro topography, the small differences where the people might have placed their sensor. And let's say we wanted to use all these to get like an average temperature for this valley. So that's an instance where we have multiple observations of this underlying state that we would like to estimate. Another example might be that we measure multiple individuals over time. So let's imagine a study where we were measuring the somatic growth of multiple individuals. And that somatic growth is an indicator of the productivity, the food availability, where those individuals are. And our interest is that food availability, and the somatic growth rates are an indicator of that food availability. And we'd like to use the information from multiple individuals. So that's another example of that. Another really common example occurs when we are sampling populations. And so let's say that the big blue here is a cartoon for our larger population. And we're going to go out in this larger population, and we are going to do 3 different studies to try to understand what the population is doing over time, whether it's increasing or decreasing over time, and how it's varying from year to year. So we have one location, and we do a mark recapture study, and kind of a small one. And then we have another location where we have a larger mark recapture study. And then we have a third location where we have a line transect. We think that all of these are measuring the same thing. They're measuring the population, but they're all a little bit different. And what we would like to do is we would like to combine all that information into our estimate of the population size. So now, like, think about that wild dog example. So now, instead of just one time series for the wild dogs, I have 3 different time series for the wild dogs. Okay, so now when we want to model this, we're going to have to write this down mathematically. What is that idea I just described? How do we write that down in equations that we can then fit to the data? So we could think of it like this. So the X is that underlying population state. And we're going to model that as before as a random walk. So this is mathematically what that underlying state is, just this. This is a random walk with drift. Now we want to say, okay, we've got 3 observations of that. What does that look like? So observation 1, so it's going to be observing the same XT, but it's going to have different variance. And it could have a different mean. So this is an index of the population. We're not sampling the whole population. It's just an index of the population. So say the whole population is, 1,000, but I'm just doing my little study in one section of it. So maybe I'm going to come up with a count that's like 200. And I'm thinking of this as like I'm sampling about a fifth of the population. So that's why there might be, this might be non-zero, kind of biased relative to this. My second one, same thing. It's measuring the same underlying state, but it has different errors because it's a different sample. And then this one is same thing. So now I want to write this as a matrix. So I can write this as one, rather than having, that whole thing filling up my piece of paper. I just want to write this as a matrix, encapsulate all those ideas. And this is how I would do that. So these are my observations. This is my YT. So it's a matrix with 3 rows, 1 column at time t. And now I'm going to multiply my state, which is 1 by 1, by this matrix, 1, 1, 1. So when I do that matrix math, you can see I'm going to have an X flowing into each of these rows. And here I have that bias. I'm going to have to fix one of them to 0, otherwise I'll have an undetermined system. And then I have my 3 errors here. Okay, so that was pretty straightforward. Now I need to think about what these errors look like and how I specify what the variance covariance matrix is for that. And that's going to mean something about how I think my the samples that I'm doing out there in nature, how they're related to each other. So in the class of models that we are talking about for the next 4 lectures, we're going to assume this is a multivariate normal distribution. We're going to move beyond that a little bit later and talk about other distributions. But for now, we're going to keep it simple and talk about these error distributions as multivariate normal. So the question is, okay, I'm going to have to say, what is the structure of that matrix? I need to be explicit about that. So let's start here with this one down in the corner. If I say that error matrix has that structure, what am I saying? I'm saying that all my observation errors across these 3 sample, so the 3 types of samples, so those 2 marker capture and that line transect, I'm saying they all have the same error variance. And importantly, I'm saying that they are independent of each other. That's a very common assumption to make about your observations, the IID. Independent of each other, and identical. Identical just means the variance is identical. So it's really important to recognize that when you see a variance covariance matrix that's got these zeros on the off diagonal, what is that is specifically saying is that these the rows, in this case, these are referring to those observations at time t are independent of each other. So if I have a high observation error in my first census, I'm not more likely to have a high observation error that year in my second census. That's what uncorrelated in this context means. A different type of diagonal matrix would be this one. And this one, they're still uncorrelated, but the variances are different. So these would be diagonal and unequal, and this would be diagonal and equal variance. So in the case of that example I gave where you've got a couple mark recapture data streams, but they're different sample sizes, and you've got a line transect, you'd probably assume that they have different observation errors, just given that they're kind of fundamentally different types of sampling designs. Definitely the line transect and the mark recaptures. Then another form that I could have is what we will call equal var covar. It's just equal variance and equal covariance. So now the variances are the same, but I'm going to allow that my observations are correlated with each other. So that means that if I would tend to have a, say, an overestimate in this measurement observation, at the same time in this other area, I would tend to also overestimate. Why might we see observation errors that look like that? Well, oftentimes when we're going out, we're estimating something in nature, it's environmental factors affect our observation error. So imagine that you are going out and you're sampling reds in a stream. Well, your observation error is strongly affected by the turbidity in the water, which is affected by the flow. So if you go out there and you do, you're sampling two different regions in the river, and the turbidity is the same because you did it pretty much at the same time, you would expect that your observation errors would be correlated because the turbidity is correlated. So that would be an example where they'd be correlated. Another example, like marine mammals, the tide level and the time of day that you're doing the sample. Those are examples where we get these correlated errors. Okay, check here. It looks like I have a chat. Okay. Let's see here. So could you give an example where we wouldn't have a one matrix? Is that even possible? Can you clarify that? What do you mean a one matrix?
Sorry, the ones matrix before like the population size.
Yeah. Well, what if there's only one underlying state, then correct. It has to be a ones matrix. You have to have one column. Well, it might not be ones in there. It could be something else. So like, what if you said that the first one has to be 50% the size of the second one? So you wanted the Y is 50% of the state and the other one is the same level. So you might then have a 0.5 and then a 1, something like that. But it is true, if you only had one state, you would have one column. It just might not be ones. But we're going to see in a moment that you don't have to assume there's one state. It might be multiple states. We'll see that in the next slides. Okay, I can't tell if this is relevant or not, but is the assumption that these data sets were collected concurrently? No, there is no assumption that they were collected concurrently. And so that in the real world, when you're setting up your models, you're going to have discussions about, okay, how are these data collected? Well, what are the assumptions we're going to make about these observation errors? Is it reasonable to assume that they have the same observation error variance? Do we need to assume that they're correlated? We often really don't want to have correlated observation errors. So we will often try to do something to make them uncorrelated. Well, such as including a covariate that would explain any correlation. Yeah, so we don't necessarily assume anything about how the data are collected and whether they're done concurrently. But what we are assuming is that what we are measuring is sampling that X at time T. So, right, so we have our estimate of the population size, say in year 1990. So the observations that we're associating with that X at 1990, they do need to be measuring the 1990. So like, you know, you could have it not concurrently, but if it was in a different year, it wouldn't make sense to say the measurement in 1991 is observing the state at 1990. But you wouldn't necessarily have to say, oh, they both occurred at noon on January 1st or something like that. All right.
Are you able to do this multi-state still, say if you miss a time period for one of the subpopulations?
Yes, absolutely. And that is one of the really big benefits about having an observation model separate from your state model. So you can just blast your observations with missing values in any kind of combination, and you're still going to be able to estimate the model. And you know what happens if you have too much missing data, you know, like any, you know, statistical analysis, at some point you don't have any power to estimate your parameters. But as long as you've got enough data to estimate parameters, missing values scattered throughout, it's going to be fine. Okay. So, oops. So the point here is just show you some examples of, so this would be your sort of errors where they are uncorrelated with each other. And then look here, this is the key one, is to show what I was talking about where I said you have correlated errors. So in this case, all of them tend to be high, all of them tend to be low, all of them, well, here they're not all high, but then here, you know, we've got a couple high together. So that's what it means where those errors are correlated with each other at time t. Okay. Okay, I'm not going to go into that. Let's just jump right here. All right, so now we're going to show, okay, in the MARSS specification, and this is actually kind of any package that you use is going to be kind of, if you're fitting these types of multivariate state-space models, it's the model specification, it's not going to be exactly the same, but kind of under the hood, it's got this same framework. Okay, so as always, what we want to do is just specify the red parameters, and we want to pass that in as a list. Everything needs to be matrices. So let's see some examples. Okay, we already went through this univariate example, so I'm going to jump past that. Now I want to jump to this example. So this one, I'm just going to have two observations. I'm not going to do the three, just to make it a little similar. So I've got two observations, and they're all observing this one state. Okay, so let's start off with our X model. So it's going to look like this. So this is a random walk with drift. We've already seen that. We've already specified that. Now what's different is that Y model. And this is what it's going to look like. So we've got our two observation time series. They're both observing this. We've got our bias there. And now we want to specify what the variance covariance matrix is. And let's say we're going to say it looks like this, which is saying that has identical variance, but they're independent from each other. So what would that look like? Okay, the U and the B and the Q, that's here. You'll notice in this example, I specified a known variance just to kind of change things up a little bit. So that Q is a known value. And here I put that in as a known value. So it's not in quotations. So it's not something we're going to estimate. It's something we're going to fix. So we just pass it in as a number. If something's in quotes, it's going to be estimated. If it's a number, then it's a fixed value. So here it's a fixed value. What is that? That is a matrix here of 1, 2 rows, 1 column. The A's, in order to have a matrix that has both numbers and characters, I need to pass it in as a list. So it looks like this, a matrix list 0, A2 in quotes, 2 rows, 1. And then the R would look like this. Do this list, and then, oh, well, something's dropped off there. So this would be a matrix with a two rows and 2 columns. So it looks like it got cut off there, the 2, 2. Okay. So that is how we would specify it. Just, if we specified everything. The nice thing is a lot of these things have shortcuts that we can use. And a lot of them will just be the default behavior. So for example, for this R, the shortcut would be diagonal and equal. And this A, usually you don't have to specify that. You just use the default behavior. It'll know how to specify the A for you. So what you're going to have to care about are things like, how do I specify the Z? How do I specify the B? That sort of thing. Okay, so let's go here. Yeah, okay, I'm going to jump through this. Let's see how I'm doing with time here. Okay, good. Yeah. Okay. So I'm going to plow a little bit more ahead onto 3, and then we'll take our break and then do our breakout and start playing around with this. So here's another example, because you were talking about, okay, would I always have in that Z matrix, would it always be a 1 with a 1 column? Well, let's see this example. So here's some multi-site data. This is an actual survey of harbor seals in our area. And the data have 7 different regions where they're doing these haul out surveys. So they go and they count the harbor seals on a haul out. The black dots are the haul outs where they counted it. And so if we were, sitting in our office, we got a whiteboard there, we're trying to think about, okay, how are we going to analyze these data? And so we're talking with the population biologist there, and they said, well, what we think is going on there is that there's a subpopulation that's kind of independent in the north, there's a subpopulation in the south, and there's another subpopulation in the Strait of Juan de Fuca. And they're kind of doing different things because the food here community is different, and you've got this kind of sill here that creates some separation between the food. And sort of the bathymetry makes this separation here. And then, really there's a lot of different things going on in the south. The Strait of Juan de Fuca, these harbor seals are heading out to the outer coast. They're really doing something different. So, talking with the biologists, they say, you need to model this as 3 different underlying subpopulations. All right. So what that's saying is, okay, we have these, that X is that underlying population process. And they're saying we need to have 3 of them. Okay, so we've got the 3. We're still going to use a random walk with drift, but now we've got 3 of them. And we're going to allow that they each kind of go up or down at their own rate. So maybe we think that, the ones out closer to the coast, they can be doing increasing or decreasing at different rates than the ones that are more inland. And then the year-to-year variability is also different. So that's mathematically how we express that idea that there are 3 different subpopulations. However, we don't think these populations are totally independent. Now that one makes sense, right? I mean, they're pretty close together. So we need to mathematically capture this idea that there may be some relationship between these 3 subpopulations. And we can mathematically express different ideas about what's going on. So we could say they're all like this. So every population could have a different growth rate, population growth rate. Or we could say they all have the same population growth rate. So all those U's are the same. Or maybe we think that the inner ones are different. They are these sort of this inland population growth rate. And the Juan de Fuca, the one that's close to the coast, that one's different. So these are 3 different ideas about how we think the population might be structured. What about the Qs? The Qs also can express some different ideas about how we think the populations are related to each other. So if we start here, this says that we think that the year-to-year variability in population growth is the same, the variance is the same, and they're independent of each other. I would probably guess that the biologist would say, well, that's not very likely. Here's a different one. This would say that they're still independent, but the variance is different. So maybe, Juan de Fuca has a different year-to-year variability. Maybe it's more variable because it's exposed to the outer coast. Exposed more to the marauding killer whales coming in, say. Okay. And then this one, we're saying, okay, they all have the same variance, but they're correlated with each other. So good years in one tend to be associated with good years in the other, but they have the same variance. And then lastly, this is the most flexible one. This says they're allowed to have different variants and they're allowed to correlate with each other in different ways. So maybe the 2 inland ones are more correlated together than the inland ones and the outer ones. Okay, how are we doing here? Okay, 2 more minutes. Okay, so hopefully that shows you how a really compact format, right here. So this is very compact. And we think of these are matrices. So this is those 3, the time series for each of the 3 populations. And this is super simple, but the structure of this U matrix and this Q matrix can capture a lot of different ideas about the subpopulation structure. And we'd like to be able to test those different ideas about how these subpopulations are structured. All right. Okay. So it is 3 o'clock. I am going to have you guys take a 10-minute break. We will come back at 3:10 and just jump back on this Zoom. So I'm not going to shut this Zoom off because I'm going to finish up this lecture and then we'll do our breakout. So I'm going to leave this going, but I'm going to stop the recording and you guys can just, I did not. Okay, great. So now we're going to look at how these observation sites map onto the subpopulations. So there's no like sort of law about how those would map onto those subpopulations. That again, how that mapping happens is what you and the biologists working on these and who are surveying these, how they think that these map onto the subpopulations. And you're going to be able to test different hypotheses about how that mapping is happening. So here's one way that that mapping might happen. So these two sites here perhaps are mapping onto that north subpopulation. These two southern ones, these gray ones, might map onto the southern population, and then the Strait of Juan de Fuca maps onto the Strait of Juan de Fuca subpopulation. So that's one hypothesis about how that mapping occurs. Not hypothesis, but that's one way you can think about how the mapping occurs. How do we express that idea mathematically? Okay, well, here are 3 subpopulations, and that mapping is going to occur in our Z matrix. So that mapping that I showed in the previous slide is this one. So this site, number 1, is that Juan de Fuca. I'm going to map that onto that subpopulation. And then I'm going to map both of these 2, onto the northern one. And then the last 2, I'm going to map onto the southern one. And you can see how this matrix is like an assignment matrix. It's assigning this one to that subpopulation, these 2 to that subpopulation, and these 2 to that subpopulation. Then I have my biases. We're not going to worry too much about that. We'll just let the software figure out how to deal with those for you. Okay, so that shows the mapping of my census sites onto the subpopulations. Now I need to think about what assumptions am I going to make about these measurement errors. So this one on the far right here would be identical and uncorrelated. So that would say the observation variances in my census sites are all the same and they're uncorrelated with each other. This would be the variances are all different and they're uncorrelated with each other. And this one would be that they're unconstrained. They could have different variances and they could all be correlated with each other. We're not going to fit models like that because it would be very difficult to estimate that. We'd need a lot more data than we have for these harbor seals. So going back to these 2, this is just one example of how I might structure it. But I don't have to say they're all different. I could say that these 2, I want these 2 to have the same variance, these 2 to have the same variance, and that one to be different. So I can structure that however I want. Just, I write down, if I wrote down these as having, these are both in south and these are in north, that would be expressing the idea that I thought the variance of these 2 sites should be the same, and these 2 sites the same. So that would be perfectly valid. That's just a mathematical way to express the idea that there's some structure to the variances. All right, so now how do I write that out in matrix form? So here it is. It's nice and compact, but all of these are matrices now. So let's focus first on the Z matrix, and let's see a few more examples. So the Z matrix. What if we thought the mapping looked like this here? So still, we've got 3, going to have 3 subpopulations, but the mapping is just a little bit different. Maybe there are the San Juan and the Juan de Fuca. Maybe those are both mapping onto this Juan de Fuca population. If that were the case, my Z matrix would look like this. This would be the mapping. These 2 are mapping onto that Juan de Fuca subpopulation. This is the only one mapping onto this northern, and these two again are mapping onto the southern. Here is a different population structure that maybe we have. Maybe it's totally different. Maybe how it works is that we have a inland population and we have a San Juan Islands population, and we have a Juan de Fuca population. So we still have 3 subpopulations, but they're structured a little bit differently. How would we express that mathematically? We would have a different Z matrix. So there's our Juan de Fuca, that's going to map onto that subpopulation. Here's that San Juan Islands, it's going to map onto its own subpopulation. And all these inland census sites are going to map onto an inland subpopulation. And then lastly, or not lastly, but another way we could think about it is that, well, you know, maybe there's just so much mixing. Maybe these harbor seals are just moving all over the place. Maybe they don't have any site fidelity at all, or not enough to create separate subpopulations. Maybe all of these inland waters are basically acting as one population, and all these sites are censusing that one population. Maybe that's how we should think about this population and think about these data. And to express that, we would have one X matrix, one population matrix, and then our Z matrix then would be a column of 1, expressing that they are all observing this one population. So I hope that gives you a sense of the way that this very compact form can express a lot of different assumptions or different ideas about how our harbor seal, or how the population is structured, and also how our data relate to the underlying population. And we can fit these different models using this mathematical construct. And then we can compare them and look at the data support for different types of structure in the harbor seals. A concrete example of this is some work that Eric and I did years ago. We were working with biologists in Baja, Mexico, and their question was about what structures the sea lion populations in the Gulf of California here. So they had data on all these different haul-outs and rookeries. They're going there year after year and sampling. And they wondered what structures the population. And they had, the biologists had these different hypotheses. One was diet, one was disease, the other one was relatedness. They had genetic data. Oops. And the other one was, well, it's just distance. The ones that are closer together tend to fluctuate together. The ones that are farther apart are more independent. So using the types of models that I just outlined, we were able to set up this big model selection study, and the structure of the models, just like what I just showed you, were expressing their ideas, the biologists' ideas about these different hypotheses. And then we could look at which structure best explained the data, was most supported by the data. And this is showing the AIC values, and the general conclusion from this study was that this simple distance model was the one that best explained the population variability in the Gulf of California in these sea lions. Okay, so the MARSS manual has 2 chapters that show examples of studies using multivariate autoregressive state space models, MARSS models, in this form. It kind of walks you through a couple of studies. It's very similar to the study we did in the Gulf of California, and how you set things up to test these different hypotheses about the structure of your data and the underlying hidden process. So I'm going to stop. Okay, so for your homework, you're going to be doing something similar to this chapter from the lab manual. So it's the chapter on MARSS models, and this one goes through using some data in the package to analyze some it's harbor sealed data. But for the homework, you're not going to be working exactly with this data. I'm going to give you some different data, but you'll do similar analyses, and it's going to be a bit more open-ended. So you'll take that data and sort of, plot it out, and then think about different ways you might structure that data, and then test some different hypotheses about the data. I haven't entirely settled on which data set I'm going to give you, but I'll settle on that by tomorrow, and I'll post that for you. So the first homework that you did, you're given specific problems, and it was quite defined. I want, okay, do this. And now we're going to be going into problem sets where it's a bit more open-ended. And you'll be trying to walk through a real analysis. And again, we, as before, encourage you to work with each other and talk about things. And if you use code from someone, say like somebody came up with a great visualization, you're welcome to use that. You maybe put in there, hey, so-and-so came up with this visualization using theirs. So that's fine. And yeah, okay. But for right now, we're going to do something more manageable, and we are going to work on this right here. So example 0 here. So let me go and open that up, and we'll start walking through that again, and then I'll get you into your different groups. There we go. Share, okay. Get rid of that. Open up. Okay, so you want this one, number 0. And since Eric's not with us, I'll be jumping between the two groups. If you have any problems. Okay, so what you'll find at the top, so you've got the things you need to load up. I'm not sure you'll need the palettes, but I put them in there just in case. You won't be able to use the forecast function today. Unfortunately, I discovered there was something that it's complaining about for multivariate data that I need to solve. But that's okay, you'll be able to plot it, plot your data. So, okay, so you run that, and you'll set up the data. There we go. So first thing you need to do is take a look at these data. So you're going to plot it. And then what you're going to do is each different group is going to assume a different population structure. And I give you the model list, so you don't have to worry about that. What you should do in the group is go through and make sure you understand why everything is specified the way it is. So kind of walk through and say, okay, what's that B? Why is that B there? Does that make sense? That sort of thing. And then you're going to go and try to tackle these questions and talk about what it means if you make that change. So for example, if you go from the observation error being diagonal, the variance matrix being diagonal, to it being unconstrained, which means you're estimating every value in that matrix. So what does that mean when you're doing that? All right. Okay. And I think that's it. If you don't get to all the questions in the, we have to have time to talk about things. So we'll get back together in 20 minutes. If you don't get to all the questions in the 20 minutes, that's fine. Just give it your best shot. We'll come back together and talk about each of them in 20 minutes.